Good morning, students. Yesterday we discussed about the microbiological control, about disinfection, sterilization, and pasteurization. Today we are going to discuss about the next topic of your textbook, which says about bacterial staining, chemotherapy, and about antibiotics. There are objectives. The objective of today's lesson is, at the end of this class, the students are expected to define the term staining and chemotherapy, explain the significance of antibiotics. The next part is about bacterial staining. It is used to identify a particular type of bacteria. Again, bacterial staining means passing of dyes during a microscopic techniques. If you remember that there are the 3 types of microscopic techniques, which are mounting, focusing, and staining. It makes a specimen conspicuous. Conspicuous means that staining makes the specimen clearly visible. For a better clarification, there are staining chemicals like crystal violet forming a purple color, methylene blue, iodine solution, hematoxylin, acetocaramine. Those are the main staining chemicals or dyes. I think you have discussed it in chapter 2 of this grade level. The bacteria that are easily decolorized and so stain red are gram-positive bacteria, and the bacteria that keeps the primary stain purple color are gram-positive bacteria. This bacteria category is based on their cell wall or peptidoglycan. Peptidoglycan is the cell wall of bacteria. So based on this cell wall, bacteria can be gram-positive and gram-negative bacteria, which have thick and thin peptidoglycan. The next one is about agar, which is keyword. A nutrient used to grow or culture bacterium in a sterilized plate or petri dish. They are also solid at 100 degrees Celsius of room temperature, but it becomes liquid when melts at 45 degrees Celsius. By the way, this is a nutrient broth mainly used to cultivate bacteria in the laboratory by using petri dish or plate. Students, please look at the staining procedures or methods which are mainly applicable in the laboratory. There are a lot of steps or procedures when you are going to stain a bacteria or other microorganisms. Let's say bacterial staining contains by having a gram staining. By the way, gram is about the name of scholar, so due to their name he named as grams stain technique. So the gram staining technique have several procedures. The first one is stain all bacteria with crystal violet or primary stain on the slide, microscopic slide. Then add a grams iodine as a mordant and leave it on the slide for 1 minute. The gram iodine and the crystal violet in the cell form a violet iodine complex. They become, and nextly again add 95% of ethanol, which acts as a decolorizer. Then the slide is ionized in water until no more color washed off. The alcohol causes the cell walls of gram-positive bacteria to retain the primary stain and appear purple. The alcohol washes away or decolorizes the gram stains of gram-negative bacteria. Finally, use safranin type of staining chemical as a secondary or counter stain. This is a basic dye that stains the decolorized bacteria into red. Leave this for 1 minute and wash off with distilled water. Finally, blot the slide dry and observe under a microscope by using focusing technique. This is how we can distinguish the staining techniques. So if you have distinguished gram-positive and gram-negative bacteria, any magnification power can be used, and the gram-negative bacteria easily decolorized and it mainly stain red. Gram-positive bacteria may remain purple, which is the primary color by crystal violet. Then to identify the details of bacteria, mainly magnification power is necessarily, which is nearest to 1000. So it is identification of gram-negative and gram-positive bacteria is necessarily for the treatment of a disease that can be affected by different types of antibiotics. So those antibiotics are drugs which kill bacteria, but they do not damage human cells. This is about the staining techniques of bacteria. The next part of our lesson is antibiotics or drug. Those are chemicals which kill bacteria without damaging the cell. If you swallow disinfectant or antiseptics, you will harm or even if killing yourself as well as the bacteria that are making you ill. But you can't take antibiotics into your body safely. They circulate in the blood and they get into all the body tissues, including the site of infection. Antibiotics only damage bacteria and do not affect human cells, as you know. Example of antibiotics like penicillin. Penicillin was the first antibiotic to be discovered by Alexander Fleming for fungal infections, mainly from Penicillium group. Penicillium is a type of fungus. From a type of fungus, Penicillium, antibiotics penicillin was produced, and it is still in use today. If you have ever suffered from athlete's foot between your toes, you will probably have used an antifungal cream to help you get rid of it. But the big problem remains disease caused by virus. Antibiotics have no effect on disease caused by virus, so they are not always the answer to our health problem. What's more, so far we have no drugs which are really effective against virus. This is something scientists and the doctors are working on all the time. As you know or you are familiar about HIV, there's no any antibiotics. Again, now at the moment, COVID-19 is also one of a pandemic disease, mainly kill through the world to all peoples without selecting any age group and so on. So mainly viral diseases are untreated by antibiotics. Look at this picture. This picture is mainly cropped from your textbook. I've cut it. So there are different types of antibiotics are found which are colored. And the next part of your lesson is chemotherapy. This is a treatment of a disease by chemicals that kill or inhibit the growth of microorganisms without harming the host at that concentration. So the examples of modern drugs or chemotherapy like chloroquine, which is used to protect against malarial infection. Again, paracetamol, as you know, used to protect against headache or severe fever. Examples of traditional drugs like cauliflower protect against tapeworm parasite. Tocrazimud, as you know, in your home, it protects against tonsil on your throat. These are chemotherapy. And as the final part of your lesson in week 2 is the additional methods that is to overcome microbes are reducing the chance of infection by isolating patients, being selected, by having personal hygiene. Again, we can overcome microbes by having a food and a water hygiene, which mean without any contamination. Waste disposal, including burning, is a main method to reduce the spread of microorganisms. Medical attention and the use of efficient latrines. Proper diet, dental safety, mandatory hand washing after and before meal, and the toilet. Those are how we can overcome microbial infection. Regular exercise and breathe in fresh, clean air. Adequate rest and sleep is also very necessary. Boiling, heating, and exposing materials to the light rays used by patients. Students, let's remind today's lesson. Today we have discussed about studying about chemotherapy, about antibiotics. Those are what have been discussed today. So thank you for your attentive listening. Bye bye.